Hi, this is Dr. L, and I'm going to show you guys what happens in day 2 of the transformation lab that we are doing with Acinetobacter chalcoaceticus. As you may recall, on day 1, we worked with 2 strains of A. chalcoaceticus, STRS and STRR. STRS is susceptible, or in other words, can be killed by streptomycin, the antibiotic. STRR is resistant to the antibiotic streptomycin. That resistance phenotype is conferred by the presence of the gene AAD2, AAD2, which encodes a protein called, let me just double check this. Sorry, I always get this one mixed up. It's an aminoglycoside 3' adenylyl transferase. So it's an adenylyl transferase protein. If you got the AAD2 gene, then you can make that protein. And then if you've got that protein, you can prevent streptomycin from disrupting the small subunit of prokaryotic ribosomes and therefore continue to make protein and survive in the presence of streptomycin. Gene, protein, phenotype. Phenotype of interest here, streptomycin resistance, which is to also say ability to grow in the presence of streptomycin. The poison doesn't kill you, the bacterium. And this is a bacterial phenotype, not a human phenotype. So on day 1 of the lab, we worked with those 2 strains, and then we took the strain STRS and we killed it using sodium dodecyl sulfate, a detergent, and heat. That killed and lysed some of the cells, releasing DNA into a solution, which we're just referring to now as the DNA solution. Those cells which were STRR that we killed, they contained the AAD2 gene. So some of that DNA released out into solution was AAD2 DNA. And so we took that DNA and we mixed it with cells of STRS, which lacked the AAD2 gene. And hopefully what happened on the plate in that little mixture is those STRS cells took up DNA from the environment, including the AAD2 DNA, and then incorporated that into their own genome, gaining that gene, and therefore gaining the phenotype of streptomycin resistance. And so what you would expect to see is what I saw on my experimental plate. So we had 4 quadrants from that. We had STRS plus DNA, and you'll notice that there's growth there. We had just STRS, and you'll notice that there's growth there. And we had STRR, and you'll notice that there's growth there. That little scummy bit is growth. And then in the DNA only quadrant, there's no growth. So the plate's a bit wrinkly because it's dried out, but there's nothing growing there. I'm gonna open this and hold it so that you can see more clearly what it would look like from the top. Okay, not that exciting, but the biology is really exciting. So what do we learn from this? Well, from the lack of growth in the DNA solution, we learned that the protocol did effectively kill all those STRR strains. And that's important, right? Because this would totally mess up the experiment if all the cells weren't killed by that protocol. So we learned that that protocol did work. We learned from the growth on the other 3 quadrants that those cells are alive and able to replicate. What we did not learn is whether or not any of them are resistant to streptomycin. We did not learn that because there's no streptomycin in this auger from the first day's work. That auger was heart infusion auger alone, no streptomycin. So we can't test for resistance phenotype using that non-selective media. So what next? Well, what we do next is we ask the question, is the resistance phenotype and therefore the gene present? And we do that by transferring these cells onto an auger plate that does contain streptomycin. They don't look any different, but we know because we put the streptomycin in when we were preparing the media. So what I'm going to do is take this plate of heart infusion auger plus streptomycin, and I'm going to divide it into thirds. I'm done worrying about the DNA solution. That was a control, and it's done. So there are my thirds. And I'm gonna label the thirds with my name around the edge. And one of them will be STRR for that strain. The other will be STRS for that strain. Notice the use of the word strain with an N and not strand, which is a common mistake. And then we're gonna do STRS plus DNA. That's gonna receive those cells that I hope were genetically transformed. So there are my labels running around the edge so that they don't obscure growth in the media. Then I'm going to get my inoculating loop out, and I'm gonna fire up the Bunsen burner. Voila. And I'm going to start by transferring the STRS cells. I'm going to flame the loop until it's red hot. And then I'm going to cool the loop in the STRS just at the edge where there are no cells. Scoop up some of the cells. Okay, I'm scooping up some of those cells. Got a little bit of growth on the tip of this. I can visualize that as a little bit of like yellow, scummy looking stuff. And then I'm going to inoculate the STRS third of this plate just by going back and forth, just in that area of the plate. Then I'm going to sterilize this loop again by flaming. It gets red hot and it burns off all the cell matter on it and it sterilizes it. Then I'm going to go in and I'm going to get the STRS plus DNA cells, the hopefully transformed cells. Go in, cool the loop, because the hot loop would kill everything, which would kind of ruin my experiment. I'm going to scoop up some cells and I'm going to transfer them to the appropriately labeled third of the plate. Wiggle, wiggle, wiggle. Sterilize again. And then I'm going to, last but not least, I'm going to transfer the STRR cells. Scoop up. Full disclosure, I totally love plating. It's oddly relaxing. And I'm going to go in and transfer those to the appropriate quadrant. Lastly, I will sterilize this loop because I don't want to put away a contaminated loop. And now I wait. A lot of things in microbiology or biology in general are setting stuff up and then waiting to see what happens. And so what I want to know is what's going to grow and what's not going to grow on this plate in the presence of streptomycin. I'll predict that the STRR strain will grow. It has the AD2 gene. I predict that the STRS strain will not grow because it does not have the AD2 gene, therefore it does not have the resistance phenotype. And I'm also going to predict or hope that my transformation experiment worked and that the STRS strain gained the AD2 gene through genetic transformation. It became transformed, it has the gene, and therefore it will have the resistance phenotype, and I'll be able to answer that question with confidence by seeing growth on that particular third of the plate. These plates will be incubated at room temperature. Overnight would be enough time, but we're going to look at them in the next lab period because it's convenient for us. And that's what we did on day 2 in this transformation lab. See you later, guys.